So what we're going to talk about today is evolution and phylogeny, specifically looking at how species are created, and how can we tell at what point those species had a common ancestor. So remember, evolution is genetic changes in a population. When we're talking evolution in a scientific sense, we're not talking about one individual, one animal, one plant. We're talking about a population. If we're talking about how an organism changes over time, through its lifetime, that's development. That's not evolution. Evolution is a population, right? So this is going to include the emergence of a species, the divergence of a species, and unfortunately the extinction of species. Right? So what we're talking about is how over years and years and years species, get an extra leg or a longer beak or get taller or fatter or change colors, get stripes. Right? Those are the things that we're talking about here. So it's a two-step process. We have genetic variation. And we've talked about this throughout the semester, how we get genetic variation in our population. We can have it through recombination, which we talked about a while back. We can have it through mutations, which we've talked about. And so the variation exists. The variation in and of itself is neither good nor bad, right? It depends on the environment, it depends on the situation, it depends on the organism as to whether or not that particular genotype or phenotype is beneficial. And so if it is beneficial over time, that genotype will increase in your population, right? So we're going to be pulling back some of those population genetic concepts that we talked about a while back. So we're going to be thinking about natural selection and migration and some of those things when we talk about changes in population today. So sometimes these phenotypes also might decrease, right? If this particular mutation or genotype causes the particular organism to not be as beneficial in that particular environment, then we're going to see it get lower over time. So populations naturally contain diversity. So most all populations have, come to a point where they have genetic diversity within that population. And scientists have often wondered, why is that? Why aren't all organisms in a population exactly the same, right? Have the exact same genotype? Why is there that diversity that exists in populations? Well, one theory is that it's just neutral, right? So we've talked about mutations and how oftentimes mutations can lead to changes in amino acid sequence that may or may not have a change on the protein, right? Sometimes we can change the DNA and it doesn't change the protein at all, right? What kind of mutations are those called when we have a point mutation that does not change the amino acid sequence? A silent mutation, right? So that's not actually doing anything. Or you could also have proteins that change the amino acid, so it might be a, what's it called when it changes the amino acid? A missense, but maybe it doesn't actually change what the protein does, right? Maybe it's just they basically do the same thing. One has a leucine, one has a valine, but the proteins are basically the same. So there's really no advantage or disadvantage. There's also the theory that it's actually actively maintained in the population, right? Maybe there's certain versions of a protein that are really important for the performance at certain temperatures versus other proteins are better at different temperatures. So having those both in the population is really beneficial because some survive better when it's cold and others survive better when it's warm. And so maybe it's actually actively being chosen or selected for in that population. Probably both of these are true in most populations. Probably there's some polymorphisms or SNPs or changes that really have no impact on your phenotype. Right? They're really not actually doing much of anything to be different. And then there's probably some that do make a difference, right? And those may be actually maintained on purpose in the population due to selection. So what we're going to spend the next little bit of time on is talking about speciation and what defines a species. So if I asked you, are a lion and a tiger a different species? What would you say? Yes. Why? They don't live in the same location. That's a good reason. They don't have viable offspring. That's a very scientific reason. They don't look alike. It's probably what most people would say, right? They look different. But species are defined as a group of individuals that actually or potentially interbreed, right? So lions and tigers don't interbreed, right? So they're two different species. Okay? So why do they not interbreed, right? We say they are reproductively isolated, right? So they're only going to breed within their species group. Now, this could happen for a couple of reasons. It could happen because they either don't choose to mate with each other or they cannot mate with each other. We call that pre-zygotic reproductive isolation. Or it could be that they could mate together, but their offspring are sterile or inviable. They don't survive. So let's think of some of these examples. So like for a lion and a tiger. Well, we mentioned they don't live in the same places, right? So lions live in Africa, tigers live in Asia. So even if they wanted to mate, they couldn't because they don't live in the same place, right? Think about, we have Asian elephants and African elephants, right? They're very similar, they look the same. I probably wouldn't be able to tell the difference, but they can't mate with each other because they live in different places, right? They're actually different species because they live in different locations. You can have situations where, so this is an image showing 945 beetles on one species of tree. Well, they live in the same place. They all live on the same tree. How are they all different species? Why aren't they all just mating with each other? Right? Well, in this case, there may be reasons why. Maybe they don't want to. Maybe they're not attracted to beetles that are different than them, right? Maybe they can't. Maybe they, are fertile at different times. Same thing with, here we have over 500 fish in a single lake of the same genus. So different species, same genus. And again, they are separate species because they do not intermate. So what are some of these reasons why these different organisms may not intermate? Well, some of these I've already mentioned, some pre-zygotic isolating mechanisms. One is habitat differences, right? Like we've already mentioned things like grizzly bears and polar bears. Right? They could mate, but as of now, their habitats are still different, right? They live in different places, so they don't. Temporal reproduction takes place at different times. This is especially important in things like plants, right, where you might have different times when they are blooming or fertile, but even things like you may have frogs, that these frogs are fertile in the early spring and these frogs are fertile in the later spring, right? So they may not mate at the same time. Same thing with the beetles, right? They might be mating at slightly different times, even of the day. Right? Mechanical, I'll let you think about that one yourself. Doesn't work, right? Behavioral, right? Maybe they don't want to, right? So think about like songbirds, right? There's lots of birds that are out in, on campus, but they're not all intermating with one another. The robins mate with the robins and the cardinals mate with the cardinals because they're attracted to one another. They have songs that they sing to attract their mates, and they're not attracted to mates of other species, right? And then gametic isolation, it's another really cool mechanism. The gametes are incompatible or not attracted to one another. So you may think, that's weird. How are gametes not attracted to one another? Well, this happens mainly in things like in the water, like in the ocean or lakes. So think about like a sea urchin. Most people know what a sea urchin is. It's not really important what it is, but it doesn't move, right? It stays right in one place. So if you're a sea urchin and you want to mate with another sea urchin, what are you going to do? You're just going to release your eggs into the ocean and hope that they find some sperm out there, right? Well, there's lots of different species of urchin. So if you release your eggs out there, you really want them to get fertilized by sperm of your same species. Otherwise, you've just wasted a lot of energy making eggs that are, not going to work. So what happens is the eggs actually secrete little chemicals that attract only the sperm of that particular species. And then once the sperm gets there, there are specific receptors on the egg that only bind to that particular sperm. So even if a sperm from a different species of urchin got to the egg, it would not be able to bind and get in because it doesn't have the proper receptors on the egg, right? So that's just one example of that gametic isolation. So then post-zygotic reproductive isolation, this would be things like when we talked about the liger and the tigon, right? We could mate them, but the hybrid is sterile, or like in the mule, right? When we mate them, it does mate. We can make a hybrid, but it is sterile. Sometimes you can make a hybrid, but it's inviable. It doesn't actually survive. Or sometimes the F1s are viable, but the F2s are not. And there's actually multiple scientists on this campus that are studying things about genetically what makes these hybrids inviable or sterile, right? How can we look at the genes in like 2 mice species and figure out, right, why are they not viable, right? Because we really don't understand genetically why that happens, right? We can just say these are 2 different species because when we try to mate them, it doesn't survive, right? So this is called the biological species concept. Now, the problem with this particular way of defining species is it only works for what? Well, that's sexual reproduction, that's true. What else? For species that are like reproducing now. Like, so if you're looking at historic species in fossil records and things, they can't really use this as much, right? Because we can guess what they mated with, but you can't really tell kind of what these historic animals kind of mated with. So when you're looking in the fossil record and kind of older species that we don't have as much behavioral and that sort of information on, they do it a little bit differently. Okay, so how do we, oh, so wait, I did have a question. Let's see. Okay, so speciation. Which one is an example of pre-zygotic reproductive isolation? So remember, a zygote is the fertilized egg. So pre-zygotic would be anything pre-fertilization, right? So pre-zygotic would mean the gametes are incompatible, so fertilization cannot occur, or differences in habitat. Right? Post-zygotic would be anything to do with a hybrid because that means fertilization has occurred. It just either is sterile or it's not viable. Okay, so how does speciation happen? Oftentimes, it's due to a geographic barrier that separates 2 groups of one species. So in this particular picture here, we see, right, here's our original population in green. Then a mountain range is the geographic barrier. So either like the mountain range, arises over time, or maybe there's a migration event that separates this into 2 groups somehow. Then there's time, right? So over time, the population on the left is going to accumulate mutations just due to random chance. There may be genetic drift, right? There may be some natural selection based on the environment that's on, the west side of the mountain range. And then over here on the right, right, we have the same thing. We're accumulating mutations due to random chance. We have genetic drift. We have potentially maybe there's different weather conditions on the east side of the mountain range. Whether there's different food, right? So over time, now these 2 species are different. And if they're brought back in together, then they're no longer going to mate. Maybe they can't mate with each other. Maybe they're not interested in mating with each other. Maybe that geographic barrier just prevents the mating, and so therefore now they're geographically isolated. Right? So this is a geographic change that prevents these 2 from mating with one another. So one example of this would be to think about, so in Africa there's lots of rhinoceros. There used to be a lot. There's not as many anymore. The white rhino is one species that is now divided into the northern white rhino and the southern white rhino, right? And as of last year, I believe it was the northern white rhino that the last male passed away, and so that species is basically extinct because now there's only 2 females left. But initially, it was one giant rhinoceros population, right? But over time, due to change and loss of habitat and loss of individuals due to poaching and things, then you end up with 2 groups that eventually became recognized as separate species because they're no longer intermating, right? So that's another example of allopatric speciation, right? A geographic barrier separating 2 groups that eventually become recognized as 2 different species. Another example of allopatric speciation is Darwin's finches, which you've probably heard about before, but it's a really great example of speciation as well as looking at how phenotypes evolve based on kind of the what's available in the environment, right? What the finches are going to eat. So we have 14 species that evolved from a single ancestral species. The ancestral species migrated to the Galapagos Islands off the coast of Ecuador, and then as additional islands arose due to volcanic activity, then they moved to these new islands, and then over time, they developed different phenotypes. So if you look at these different finches, right, if you look at their beaks specifically, you can see there's differences in their beak shape, and the beak shape is directly related to what they eat. So when there are seeds on the ground, the birds have the short, fat beaks because that's easier for them to pick up the seeds and break them open on the ground. Versus if there are cactus fruits, which is where they get their food, then their beaks need to be long and pointed to get into those cactus flowers, right, to get the seeds out. So what they're eating has, over time, determined what physical features that they have evolved. Right? So these are all different species that came from a single ancestral species a long, long time ago. Right? And to bring it a little bit full circle, this actually is all dependent on variation in gene expression of a particular gene. It's expressed in a much wider range in the wide-beaked ones and a much narrower range in the pointed-beaked ones. But that's not, I'm not going to ask you about that, but it's just kind of making that connection. So there's a picture of the Galapagos, and we can see as the number of islands increases, which is in green, the number of finches species increase. So basically every time a new island arose, some finches migrated there, and then over time they evolved to have features that were specific to that particular island. So that's allopatric speciation. Questions about that one? Makes sense, right? Well, what about those beetles on the tree, right, in the rainforest? They're all in the same place. They're all in the same tree, right? How are they reproductively isolated? Well, one example of that would be an example of sympatric speciation, where we have a single interbreeding population that can evolve to be multiple species even without a geographic barrier. So how in the world could that happen? Well, basically, in this example, we have the apple maggot fly that is now currently kind of becoming two different species due to very strong disruptive selection, right? Selecting for two very different sets of genotypes in this particular location. So in this case, the apple maggot fly used to eat a lot of hawthorn tree fruit, or some of them ate hawthorn tree fruit. Well, originally they all ate hawthorn tree fruit. Then a mutation arose, and some of them now can eat apples. So these apple trees and hawthorn trees are all in the same forest, right? But now some flies can eat apples. Well, if you're a fly, who are you going to mate with? The people that you like hang out with, right? So the people on the apple, or the flies on the apple trees are going to mate with flies that are on the apple trees, and the flies on the hawthorn trees are going to mate with other flies on the hawthorn trees. So we're starting to get two groups, right? Two separate groups of flies. Now they're not necessarily two species yet, they're just kind of two different groups that are kind of mating within each other. Well, turns out, if you know a little bit about these flies, they lay their eggs in the ripening fruit, which, yeah, but the ripening fruit is a little softer, right, than the final fruit, so they lay their eggs in the ripening fruit. Well, it turns out that apples ripen about 3 weeks sooner than hawthorns, whatever those are. So the apples are ripening sooner. So over time, all of those flies that are hanging out on the apple trees have evolved such that they are mating 3 weeks earlier than all the flies on the hawthorn trees. So their mating time is changing because it makes a lot more sense for them to be mating at a point where they can lay their eggs in the growing fruit, and the fruit is developing at different times, right? So now that we have these two groups that are mating with each other, they're continuing to accumulate mutations and changes that make them more suited to their specific kind of tree that they're growing on. Now, speciation is not yet complete in this case because we still do have some mating between these two groups, right? So speciation in many cases is a process, but they're pretty isolated, but not completely isolated. Okay? Does that make sense? Pretty cool, right? Another example of how sympatric speciation can happen, right? This might look vaguely familiar. We talked about this before. Right here we have two grasses, right? So you can have multiple grasses in a field or in your yard, right? Grasses and weeds, and they all kind of coexist together. And then you could have two grasses that come together to create a hybrid. Now remember, this hybrid is sterile. These are two different species that came together to form a hybrid. But if a non-disjunction event occurs, now we've doubled all of the chromosomes, and now we have a fertile hybrid. So now we have two, potentially three now species all in the same area. But that's also an interesting question of, like, when do you call a species, and when is it a hybrid and when is it not, right? So one thing I mentioned a while back is that the red wolf for a long time was seen as a hybrid of the gray wolf and the coyote. Right? So, and then the question is it a hybrid? Is it a species? What are we going to call it? If it's a hybrid, then does it deserve the same endangered species protections of the Endangered Species Act, or is it a separate species? And actually, now that we have released so many red wolves back out into the wild in North Carolina, because this is one of their primary habitats, they're really concerned actually about the red wolves mating with coyotes and further diluting kind of their species into kind of a more hybrid red wolf coyote hybrid, because coyotes have taken over a lot of their habitat because they were basically almost completely, I think they were completely gone from the wild, and now we've reintroduced them. But anyway, that's a side story. Okay, species, yay. Questions about speciation. Really interesting topic to think about. Okay. So the next thing we're going to talk about is how do we understand the relationships between these species, right? So not all species can we kind of watch them evolve into two species. Can we watch speciation occur? Most of the time we're looking at species that exist and try to infer past relationships. Like, at what point did these species have a common ancestor, right? And that's what phylogenetics is. It's studying the relationship between individuals or genes or alleles based on characteristics. So we can reconstruct these phylogenies by inferring relationships about present-day organisms. So you can say, okay, when I compare a human and a dog, I can make a guess as to how long ago did they have a common ancestor, and the human and the dog had a common ancestor more recently than the human had a common ancestor with the kangaroo, right, et cetera. The tree of life, you can see here, it kind of starts here and branches out. It's a reconstruction of the history of speciation and extinction events for all the life on Earth using phylogenetics. Right? If 4.5 billion years of the Earth's history were condensed into 1 year, humans have only been around for about 24 minutes. Right? So there's a lot of things that have come and gone before us. There have been multiple mass extinctions that have happened over the last few million or billions of years. Those are the dinosaurs there. And then these are humans when we showed up. Right? Okay, we're not going to do anything this major. We're just going to stick with small trees. And really what we're looking at is we're using these trees to tell us relative relationships between organisms. Okay, and we're going to talk about how to build a tree and show these relative relationships. So, what are different types of evolutionary change? One thing to remember is that all species are constantly changing over time. All populations are continually changing over time. So when you think about things like alligators or sharks that have been around for millions and millions of years, they're not the same as when they were here prehistoric times, right? The alligators of today look different than the alligators of millions of years ago, right? They have evolved over time. Even though the species still exist, they're still evolving. That's called anagenesis. That's the horizontal lines here. They indicate anagenesis, or change over time, right? So they're continually changing over time. Cladogenesis is when your tree branches. So when the tree branches, that means that we've had a speciation event, and now this one species has now become two species, right? The point at which the speciation event occurs is typically called a node. So these branches lead to more diversity. So we have more species existing at the same time. So if you were going to create a phylogenetic tree to try to make comparisons between organisms and try to determine which ones are more closely related or had a more recent common ancestor, what would you look at?
Maybe like the percentage DNA we have that's the same. I know that's kind of how we compare like humans and like other primates and stuff like that.
So that's what we do now is we look at DNA. What did we do before we had DNA to look, or before we could sequence DNA?
It was more of just looking at what kind of features everything shares.
So looking at morphology, just kind of looking at how things are similar or different. And that's still a lot of, again, what's done with many things in the fossil record that we don't have DNA for. But what we typically do now is look at DNA sequences, right? So we're going to compare the DNA of humans and primates, or humans and dogs, or cats and chickens, to see how many changes there are if we compare those two and kind of make a prediction as to when they had a common ancestor. For things that we don't have DNA sequence for, we still have to use things like morphology, potentially chromosome structure and things to make those predictions. Okay, so homologous sequences are related evolutionarily. So this is kind of what we're basing our idea on, is that when we have sequences that are very similar, we think that they came from a common ancestral sequence. So how does that occur? So let's say here's the ancestral fish. He has gene A, right? So gene A may be very important. Maybe gene A is really important for making the spinal cord of this fish or something, right? Then somehow this gene is duplicated. So now we have 2 copies of gene A. And now the second copy of gene A is accumulating a lot of mutations so that it looks very different than the first copy of gene A. And it turns out that the second copy of gene A now causes the fish to have stripes. So these are called paralogs. A1 to A2 are called paralogs. So these are 2 genes in the same species that are homologous or came from a common ancestral gene. Now let's say a speciation event occurred, right? Somehow these 2 groups are separated. We have 2 species of fish. And over time, right, this species on the left, it's evolving and changing, but it's basically, let's say it's just looking very similar, but know that evolution is still occurring here on the left. And on the right, evolution is also occurring, but it's different changes are happening, different things are being selected for. So now we have 2 different versions of these genes, B1 and B2, and now the fish kind of has spots. So B1 and B2 are orthologs to A1 and A2, right? So again, they came from a common ancestral gene, but now they're in different species. In humans, we have a lot of examples of these gene duplication events. One is the globin gene family. So the scientists believe that there was a primordial globin gene that was duplicated. One of those duplicated versions evolved to become myoglobin, right? And then the other globin gene duplicated to become the alpha globin and the beta globin gene cluster. Right? And then the alpha globin gene cluster had multiple duplications to become all these versions of alpha globin, and then we have all these versions of beta globin. Right? So all of these different globin genes are really important for our development and what makes humans humans. Right? Over time, we've had all coming from this ancestral globin gene. Another example of gene duplication that I've already told you about is the Hox genes, right? So if you look at Drosophila, they only have 1 copy of this cluster, versus in mammals we have 4 copies of this cluster, right? And so this cluster has duplicated multiple times over evolutionary time in what we ended up coming into see in mammals. And also the last Hox gene, Hox9, has also duplicated multiple times. Right? And so they've diverged in their abilities now that we have all these extra Hox genes can do extra things in mammals and in humans. But the really cool thing about the Hox genes is that they still have very similar roles in all of the organisms that they are present in that, Hox1 is going to be the one that's in the most anterior for specification, and then HOX 9 through 13 are the most posterior for cell fate specification. Questions about gene duplications? Okay, so how can we use these duplication and divergence events to set up our phylogenies? Well, one thing that we can do is assume that your genome mutates at a constant rate, kind of on a population level, right? There's changes are happening at a constant rate. If we assume that mutations are random, and we're not accounting for any sort of like mass radioactive event or something like that, right? We're just having kind of a constant mutation rate. Different parts of the genome are going to accumulate mutations at different rates. And we can look at those rates and use those to help us make these predictions. So the highest mutation rates in your genome are observed in the regions that have the least effect on gene function. What does that mean? Well, if you look at this chart here, what is the lowest number that you see in the chart? What is it? What kind of sequence is it in? The coding sequence. Coding sequence are sequences of DNA that code for proteins. So you can imagine mutations in those regions have the potential to be much more damaging, right? If you're going to screw up a protein, that's potentially going to be a problem. So those mutations are going to be maintained or evolve at a much slower rate, right? Because the likelihood of you screwing something up is much higher. Versus if you look here at the pseudogenes, that's a really high number. A pseudogene doesn't actually do anything. So we can change that up all we want to, right? Mutations can accumulate there at a higher rate because it's not actually really that important for the function of the organism. So the more constraints on a sequence to provide function, the fewer changes are tolerated, meaning that if it's an important part of the DNA, it's going to make a protein or do something really important, we can't really screw that up very much. So we're not going to tolerate many changes. Now, if you see the 5' flanking region has a much lower mutation rate than the 3' flanking region. The 5' flanking region, remember, is what's upstream of the transcriptional unit. That typically is going to have your promoter, your regulatory elements. So those are going to be pretty important. So those are less likely to change. The leader and the trailer that you see here, these are the untranslated regions. And then we have introns, and we have pseudogenes. So you can look at this chart to kind of figure out what are the regions of a particular gene or the regions of the genome that are more able to be mutated over time. Now remember, mutations are just happening, but some of them are going to be able to remain in the population, and some are going to be lost because they're detrimental. So this is just a picture showing you basically what I just told you. So it's just the chart in picture form. So here we have pseudogenes have the really highest mutation rate, right? We didn't really talk about the synonymous substitutions. That's the same thing as a silent mutation or a missense mutation that doesn't change the amino acid structure, right? So you change the amino acid, but the protein doesn't change. So that's what a synonymous substitution would be. Non-synonymous substitutions are going to change the amino acid or change it to a stop codon. Mutations in the intron, those are pretty well tolerated because the intron's mostly not important stuff. There is some important stuff, but not much, right? But the lowest mutation rate are in the exons. I did want to mention here that sometimes you can have introns in your untranslated region, and that's not super evident from this picture. All right, questions about mutation rates. So why is this important? Why do we care about mutation rates in different parts of the genome? Well, if we assume constant mutation rates, what we can do is compare two sequences and say, ooh, here's a sequence from a cat and here's a sequence from a chicken. How different are they? How many changes are there between the two? How many mutations have happened between those two genes there? And then we can use that to make a prediction backwards to figure out when did they have a common ancestor. And we use this as the molecular clock, right? So based on the assumption of a constant mutation rate, we can take the differences in the genetic information of present-day organisms to infer back to when they had a common ancestor. All right, does that make sense? Now we're going to make some trees. Okay, so we can use this molecular clock to create a phylogenetic tree. A phylogenetic tree is a graphical representation of the degree of similarities between OTUs, which OTU is the technical term operational taxonomic unit, meaning it doesn't have to be a species. It could be that we're comparing just gene sequences. It could be that we're comparing strains of a virus or bacteria. So it doesn't have to be different species. But basically, right, it's a graphical representation to show us the relationships between these different organisms. So there's different ways you can make trees, and if you spend more time in evolutionary genetics types courses or evolution type courses, you may talk about different types of trees. These are two types of trees, an unrooted tree up here at the top and a rooted tree at the bottom. So at the unrooted tree, we can figure out the molecular distance or the evolutionary distance between A and B and between C and D and between the A and B group and the C and D group, right? In a rooted tree, not only can we tell the distance or the changes that have happened between A and B, but we can also figure out when the divergence happened or when they had a more recent common ancestor. So if you look at C and D, right, they had a more recent common ancestor than B did to C and D, right? So you can tell kind of when the divergence happened. Typically it's rooted because we have an outgroup, which is a group that we know is the most distantly related to the other groups. And that can what we call root our tree. So if we know that this particular organism is the most further away from the rest of them, it gives us a root for our tree. We're going to mainly focus on these rooted trees here. So if you look at your unrooted tree, like we know the distance between A to B, but it could be that the B species split off first, or it could be that the A species split off first. We don't know. Both of these trees are compatible with this unrooted tree. So depending on what you're looking at, an unrooted tree might be more accurate, but we're going to, again, stick with the rooted trees on the right. Okay, here is an example of a phylogenetic tree. So this is a phylogenetic tree of the genus Equus, which has a bunch of different types of zebras, and it is related to the horse, but the horse is our outgroup, right? It's more distantly related to all of these here. Now, what we have here is, here are our different organisms. These horizontal lines are indicating relative evolutionary time, okay? So when we have this here is a much shorter line than this one here, meaning that these two had a common ancestor more recently than the other two, these two. Oops. Here. So the donkey and the Somali wild ass had a common ancestor more recently in evolutionary time than the Tibetan wild ass and the half-ass, right? So their common ancestor is more distant in evolutionary time. Then the common ancestor to these two and the common ancestor to those two had a common ancestor here, right? Each time that these come together, it's called a node. Okay, and the nodes represent where there's a common ancestor, right? And then, again, the horizontal lines represent relative evolutionary time. If they're shorter, it's a much more recent, and if it's longer, that was a longer span of time. Okay? So let's see. If you look at this phylogenetic tree, which two species are the most closely related? Okay, so of those four animals, can we rule one out? The horse. The horse is our out group. So we know the horse is not going to be one of the two species that are most closely related. So what we need to do is look at the mountain zebra, Grevy zebra, and Grant zebra. So if we look at the mountain zebra, Grevy zebra, and Grant zebra. So we're basically going to go back. So Grant zebra and the quagga share a common ancestor here, right? Then the quagga and Grant zebra's common ancestor diverged from the common ancestor to Grevy zebra here. And then the common ancestor to those three diverged from the ancestor to the mountain zebra here. So the most closely related are going to be the Grevy zebra and the Grant zebra because they have a common ancestor that is more recent than the common ancestor with the mountain zebra. Does that make sense? Questions about that? Okay, so we can also use this for viruses and things of that nature. So this is an image. It's actually from the 5th edition of the book. They took it out of the 6th edition. But this is based on a true story, so this actually happened. There were several patients from one dentist in one community that developed AIDS. And it turns out that the dentist also had AIDS. And so the question was, did these patients contract HIV from the dentist or just from the community? And so what they did was they took 2 samples from every individual. The HIV virus mutates very rapidly. So you could have, even within your body, multiple versions of that particular virus. So they took 2 samples from each person and sequenced the envelope gene. So they sequenced 1 gene in those samples, and then they created a phylogenetic tree based on sequence divergence. And if you look at this phylogenetic tree, we see that up here at the top is the dentist here. And he is much more closely related to patients C, A, G, B, and E than they are to any of these local controls, which is what the LC is. However, patients F and D are more closely related to the local controls than they are to the dentist. So actually it turns out that malpractice insurance did pay out claims to those individuals A, B, C, E, and G, which were most likely infected by this particular dentist in this particular case, right? So you can use this sequence divergence not only to kind of figure out what animals or species have the most common ancestor, but also strains of viruses. You can look at versions of bacteria. I actually went to a talk recently from a scientist who works at the FDA, and they're doing sequencing to try to help them identify sources of outbreaks, right? So they're having all these different samples from different plants and different places that packaged food and whatnot, so that if there's an outbreak of like E. coli, then they're going to sequence some of that E. coli and see if they can figure out, can they match it to one of the E. coli samples that they have from these various plants in different places throughout the country to figure out kind of where that outbreak is originating from, right? So looking at the sequence divergence to try to figure out where it's most closely related to, and can you identify the source, right? So there's lots of different ways you can create these trees. If you go further in evolutionary genetics or in evolutionary biology, you may do trees in some of these other manners. We are going to focus on the distance approach. There's also the parsimony approach and the maximum likelihood or Bayesian approach. We're not going to worry about those. I just want you to know that there are other ways to do this. So basically, the distance approach is we're going to look at the similarity of the organisms, and we're going to look at sequences by using these sequences to infer similarity. And we're basically going to compare the sequence, the DNA sequence of organisms, and see how different they are, and use that to create our phylogenetic tree. So we're using what's called the unweighted pair group method with arithmetic mean. No, you don't have to remember that. It's a relatively complicated phrase, but it's a simple method. Basically, we're going to look at the differences in the DNA sequences, and it doesn't necessarily require an outgroup in the way that we're calculating it. We're going to use OTUs. Most of the examples we are using different species, but you could just use different strains of a virus. You could use different genes, that sort of thing. So the numbers are calculated from the differences in the DNA sequences based on sequence alignment. And this is important. We have to line up the sequences to figure out how many differences there are, right? And typically computers will do this for you, but just an example of how this would work. So let's say we have two sequences from two different species, right? Sequence 1 and sequence 2, and we're going to line them up. Well, you could line them up as you see at the top, and if you do that, there are 5 differences between sequence 1 and sequence 2, right? But actually, if you just insert a hole there, or say that that T was inserted, right, have a kind of a space there, then we can align them more closely, and now we only have 2 changes between those 2 sequences. So when we do a sequence alignment, we're going to try to minimize the differences. Now, in reality, we don't actually know, right, all of the evolutionary changes that occurred, all of the molecular changes that occurred from the common ancestor to these 2 sequences. But we're going to assume the lowest number to kind of minimize our predictions, right? Instead of kind of saying, oh, there was hundreds and hundreds of changes, we're going to minimize that, right? Okay, so what are we going to do? How are we going to calculate distances? And so yes, in this unit 5, we will be doing math again, doing some calculations so for those of you that missed that, brought it back. All right, so we're going to do this by basing it on the fraction of nucleotides that are different. So how many bases are we looking at here of sequence? 10. How many bases are different? We're using this one here where we have the minimum number of changes. So 2 out of 10 is 0.2, right? Now, to make things simpler, this isn't working. We multiply this by 1,000 to make the numbers easier to work with. I honestly don't know why they multiply by 1,000, but I was told that that's what we do, so we do it. But it does make it easier. You don't want to work with 0.2 and 0.8, right? So 200 is a much easier number to work with. Okay, so now we know that our, quote, differences between sequence 1 and sequence 2 are 200, okay? But the really important thing to remember is that what we're trying to do is try to figure out how long ago did those 2 sequences have a common ancestor. So the common ancestral species was different than both sequence 1 and sequence 2, right? So if we say there's 2 changes in this region between sequence 1 and sequence 2, we could assume that each sequence had 1 change, so that the ancestral sequence probably had one of the sequence 1 versions and one of the sequence 2 versions. right? So when we try to look for that ancestral sequence, we're going to say that it's half of the changes from one sequence and half of the changes to the other sequence. Does that make sense? Kind of, sort of? All right, let's try to make a tree. Okay, so here's our chart. I went ahead and calculated all of the numbers for you. You will have an example of problem session next week while we'll actually do the calculations and figure out the numbers, the percentage distances or differences, between the DNA sequences. All of the numbers, I will go ahead and tell you, all the numbers that we're using for this section of the class are completely fictional. I made them up to make them easy to work with. However, I did try to maintain evolutionary relationships that are predicted. So the relationships that we're drawing on our charts, which one has a more recent common ancestor, that is maintained, but the numbers are completely random. Well, not random, but I made them up. Okay, so let's see. If you look at the chart here, which two species do you think are most closely related based on your numbers? So remember, the numbers are calculated by taking the percentage of differences between the sequences in these two organisms. I'm going to rewrite this. Okay, so if we're looking for the percentage of differences, if they're the most closely related, are we looking for the smallest number or the biggest number? The smallest number, right? The lowest percentage of differences. So it would be the bottlenose dolphin and the narwhal, right? Because that number is the smallest number that we see on our chart, right? It's 20. All right, so if we're going to create our phylogenetic tree, what we're going to assume is, so the 20 between the bottlenose dolphin and the narwhal, I mean, technically that meant that there were 2% of differences between those two sequences. You can also just think of it as representing like 20 changes. It's not exactly what it means, but it's kind of the same idea. Okay, so if we just think of it as like there's 20 changes in the sequence from the narwhal to the bottlenose dolphin-ish, right? Well, that means that from the bottlenose dolphin to the common ancestor, there were probably how many, quote, changes? Say 10, right? And then from the narwhal to the common ancestor, there were 10 different changes, right? So that when we compared the two, there's actually 20 differences between the two, but they're only 10 different each from that common ancestor. Does that make sense? Right? So if we started in the middle here, and we had two species, and each of you took 10 steps in that direction, you would each be 10 steps away from me, the common ancestor, but you would be 20 steps away from each other. Does that make sense? Okay, so that's how we're going to put it on our phylogenetic tree. All right, so here we have our bottlenose dolphin and our narwhal. We're going to say they're each 10 away from that common ancestor, but they're going to be 20 away, right, from each other. Right, so then the question is, what are we going to put next on our chart? Well, the bottlenose dolphin narwhal, we know that they have a common ancestor. So what we really want to figure out is which one, the sperm whale or the humpback whale, is most closely related to this common ancestor of the bottlenose dolphin and the narwhal. So in order to do that, we're going to average the calculated distances from the bottlenose dolphin to the sperm whale or the humpback whale and the narwhal to the sperm whale or the humpback whale. All right? So let's do that really quick. So if we have the bottlenose dolphin narwhal together to the sperm whale, so bottlenose dolphin to the sperm whale is 100. Narwhal to the sperm whale is 150. So this is going to be 100 plus 150 divided by 2, 125. So then bottlenose dolphin narwhal to the humpback whale. So bottlenose dolphin to the humpback whale is 200. Narwhal to the humpback whale is 150. So 200 plus 150 over 2 is 175. But then we also have the humpback whale to the sperm whale, which is how much? That's the same, right? So if we humpback whale to the sperm whale is still 200. So in our new chart, right here, what's the smallest number there? 125. So that tells us that the sperm whale is more closely related to the bottlenose dolphin narwhal common ancestor than the humpback whale is. And again, if the sperm whale has 125, quote, changes from the bottlenose dolphin narwhal group, then how many changes or how many steps away is it from the common ancestor? So again, if you have a common ancestor and it splits, right, if the sperm whale is 62.5 steps away and then the bottlenose dolphin or the narwhal is 62.5 steps away, they're both 62.5 steps away from the common ancestor, but they're 125 steps away from each other. So if this bottlenose dolphin or narwhal is 62.5 away from this common ancestor, what number should go on this line here? 52.5. Now I didn't give myself a lot of room, but over here, right, so we know our last one is our humpback whale, right? So in order to figure out the distance from the humpback whale to the common ancestor, we need to figure out the average of the distance of the humpback whale to the sperm whale, narwhal, and bottlenose dolphin. Because we want to figure out what is the distance for the humpback whale, right, to this common ancestor, to all three of those. So we're going to go back to our original chart up here at the top. Say the humpback whale to the sperm whale was 200. The humpback whale to the bottlenose dolphin was 200. And the humpback whale to the narwhal was 150. We divide that by 3. That gives us 183.3. Now remember, to go from the humpback whale to the common ancestor, we need to take this 183.3, 183.3 and divide it by 2. questions so far? I'm going to walk through it again on a laptop. All right, I'm going to pull this back up. Okay, so just to kind of walk through the whole thing again, the most closely related are the narwhal and the bottlenose dolphin, right? So if the narwhal and the bottlenose dolphin are 20 apart, they're each 10 apart from that common ancestor, right? Because if the common ancestor is in the middle and they each walk 10 steps in either direction, they'd each be 10 from the common ancestor, but they would be 20 from each other, right? Then to figure out which species is most closely related to that common ancestor of the bottlenose dolphin and the narwhal, we're going to have to do some averaging. So what we did was we took the bottlenose dolphin to the sperm whale and the narwhal to the sperm whale and averaged that, and that gave us 125. And then we took the humpback whale to the bottlenose dolphin and the humpback whale to the narwhal, and that gave us 175. So on this chart, the smallest number was 125. So that tells us that the bottlenose dolphin narwhal common ancestor split from the sperm whale, right, in more recent time than the humpback whale. Okay? And again, we can say that we think that it's on the average is there's 125 quote changes or steps between the sperm whale and either the bottlenose dolphin or the narwhal, which means there's going to be half of that, 62.5, from the sperm whale to the common ancestor, and 62.5 from either the narwhal or the bottlenose dolphin to the common ancestor. And then if we're going to add the humpback whale, so the humpback whale, we have to average everything else from this original chart here. Right? So that's really important. When we do this average to figure out what do we think is the distance, approximate distance from this common ancestor to the humpback whale, we have to go back to the original chart here and take our 200, 200, and 150, average it to get 183.3. So that tells us that there is approximately, right, 183.3 steps from the humpback whale all the way to the sperm whale, right? But from the humpback whale to the common ancestor is approximately half of that, which is 91.7. Right? So again, all of these numbers are kind of a relative expression of change. Right? So when you look at this picture, you can see that more recently in evolutionary time, the bottlenose dolphin and the narwhal had a common ancestor. More historic to that, there was a common ancestor that actually eventually became the sperm whale, bottlenose dolphin, and narwhal. And then even further back from that, there was an organism that was the precursor to all of these. Okay? Yes?
How did you get 29.0?
So that would be to go from this common ancestor here all the way to the sperm whale needs to be 91.7. So we take 91.7 minus 62.5, and that gives us the 29.2. Sure. What else? So again, these numbers are somewhat kind of relative measurements of change. You're going to do a couple different examples of creating these phylogenetic trees in problem session next week. So I know we have the exam next week and you're going to be studying and working really hard on that, but don't skip out on problem session because it really is helpful to practice doing these and making sure you really understand kind of what these numbers represent and what these different parts of this tree represent. Okay? Any questions? Okay. I'm actually just going to stop here for today, and we'll start on cancer genetics next week.